Okay, let's get started. Welcome to the last invited talk from this semester. It's a pleasure to introduce Professor Noeska Smit from University of Bergen. Noeska is an associate professor in medical visualization at University of Bergen in Norway. Besides that, she is also a senior researcher at the Medical Imaging and Visualization Center at the Department of Radiology at Auckland University Hospital. And her research interests include model-based visualization for surgical planning and education, and also interactive visualization of medical imaging data. And Noeska also received the Dirk Bart's Prize for Visual Computing in Medicine in 2019, so this year. And Noeska did her PhD at Delft University of Technology in the Netherlands. And now, in the next probably 45 minutes, Noeska is going to talk about her work on medical visualization, how we're going to visualize the invisible. Thank you. Noeska, go ahead.
Thank you very much, Mark. And thank you also for inviting me for this talk today. This is a real honor and a great opportunity. Thank you so much. Yeah, so you already gave a great introduction to who I am, but I'd like to dig further into the past just a little so you know a bit more about my background. Because before I studied computer science, I actually worked and studied in the hospital to become a licensed radiographer. And a radiographer, if you don't already know, that's the person in the hospital that's responsible for taking the medical imaging scans, like CT, like MRI, and also for taking the X-rays. And part of this job was for me to sometimes play dress up a little. So this is me about 15 years ago. When patients were contagious, we had to go into the rooms with these mobile X-ray units. But after 3 years of this work, I got kind of tired of this and working with the patients so directly. So I decided to study computer science at the University of Delft. And here I really specialized in computer graphics and visualization. And my beginning goal with this was to become a game developer, actually. But in the end, I found out that there's also this field called medical visualization where my background really helped. So in the end, I specialized in medical visualization. And here I also did my PhD. And even though I thought I would never be dressing up in such funny outfits again, also during my PhD, I got this opportunity again. So even 15 years later, again, I was in the same clothes attending a surgery. Currently, I'm indeed an associate professor in the Bergen Visualization Group, and I have a 20% position as a senior researcher at the Medical Imaging and Visualization Center, which is also at the Department of Radiology in the hospital. Yeah, so with the audience here, I don't think I need to explain to you what visualization is or to convince you of the use of it. But in general, of course, with visualization, what we want to do is we want to go from data, models, and simulations to insight for the users. And in my research in particular, I'm focusing on medical data. And when you're looking at the field of medicine, in recent years, there's been an exponential growth in the amount of data that's being generated. And this also leads to an increased complexity on the analysis. So the more data you have, the more difficult it will become to analyze it. And this places then higher demands on visualization to make decisions. And here's some example also of this exponential growth in gene data, which I know that your group is working on a lot. In general, in medicine, there's this thing called P4 medicine, which is a sort of medical paradigm shift. And the aim is here to go from medicine as something that is more aimed at diagnosis and treatment into something that is more predictive and preventative, personalized, and also participatory. And the goal with this is to shift from a reactive way of operating. So once a patient gets sick, you treat him for it, to a more proactive approach where you can identify diseases even before they happen and do something about them earlier on. And to make all of this possible, there's been richer data acquisition than there has been before. And we see this also back in medical visualization, where traditionally we did more direct visualization of anatomical imaging data, for instance, from CT scans and MRI scans for diagnostic treatment planning purposes. Now, if we want to aim at prediction and prevention, we need more advanced techniques. And these techniques can be beyond what is available from medical imaging scans directly. So here's an example of such medical imaging data, in case you've not seen it before. This is an MRI scanner. And what comes out are medical imaging slices like this. So here's a brain scan in the MRI. And these days, it's no longer just one brain scan that comes out, but very many. So for a single patient, there may be multiple of such scans. So you're no longer dealing with one 3D volume, but with multiple of them. And the point of this multimodal imaging, as they call it, is that each of these types of contrast provide additional data and insights into the brain in this case. So here on the left, you see a CT scan, which provide a certain type of contrast where the bone is very well visible. And on the right, you see several different types of MRI acquisitions. So a T1 weighted imaging scan, T2, and you'll see that, for instance, in T2, the water in the brain is really highlighted. While in T1, you can clearly see the differences between the white and the gray matter. Now, this wealth of medical imaging acquisitions is meant to provide complementary information from different modalities. And then visual integration of these modalities can be very useful to further aid diagnosis and treatment planning. But also in case of prediction and prevention, if you have these more detailed data sets, you can do much more with them. I'd also like to show you my brain. So there are many like it, but this one is mine. So this is a normal MRI scan of the anatomy of my brain. You may notice that the jaw is missing. This is because I have braces and they drain all the signal. It's not that I actually do not have a jaw. Anyway, this is one scan, and there's 188 slices of anatomical data. Now, for this, we don't need very advanced medical visualization techniques because we can simply scroll through them and get a good mental model of what the data is like. These days, however, they can also do functional imaging. And in functional MRI, you have 100 scans added of 34 slices, so lower resolution, of functional data. And if you need to look at these one by one and scrolling through them, it becomes a bit too much. And here, interactive visualization can really help. So what I'm showing you here is 101 scans visualized in one in animation. So we're using the anatomy scan as context, used to extract the surface of my brain. And then we're using these 100 functional scans as an animation to show the activity over time. So what I'm doing here is I'm finger tapping my left hand and then I'm finger tapping my right hand. And in the scanner, I was doing this first on the left and then on the right with a break in between. Now, using interactive techniques, we can also do more with the data than just play it back as an animation. We can do time warping. So what I'm showing you here is after the activity on the left has occurred, I'm brushing over it with my mouse and warping back the time to align it with the finger tapping on the right. And in this way, it becomes possible to explore data in ways that are not possible by just looking at the medical imaging scans directly. So here's the time warping taking place. And now you see both regions of the brain highlighted at once. In general, in model-based visualization, what you're doing is you're enriching measurements such as you have for medical imaging data with models or simulations. And a familiar example of this is when you're looking at weather data. So to predict the weather, just measuring the weather is not enough. What you need to do is you need to add simulation models to add more information to it so you can come up with a better prediction. And today, in today's talk, I'd like to discuss a concrete example of using model-based visualization to enrich medical imaging data. And this project was done a while ago, and it was a close collaboration between the Leiden University Medical Center, the Department of Anatomy there, and also the Department of Surgery. And then on the other side, the Delft University of Technology, where I was working at the time in the computer graphics and visualization groups. Now, this project in particular dealt with a certain disease, and that disease was rectal cancer. So colorectal cancer is the third most commonly occurring cancer in men and the second most commonly occurring cancer in women. So it's quite common. And in the Netherlands alone, there's over 4,000 patients every year for this disease. And the 5-year survival rate for this disease is only 50%. That means that 50% of the patients die within 5 years. Now, luckily, there is a treatment for this disease, and it's a surgical treatment called the total mesorectal excision. And in this procedure, what they're doing is they're taking away the part of the rectum that contains the tumor, as well as all the fatty tissue around it, the mesorectum. So this is quite an invasive surgery, but it has drastically improved the survival rate of the disease. Now, unfortunately, there are some drawbacks to this surgery, and these are the surgical complications that occur as the surgeons are doing it. And these are urinary incontinence in 34% of the cases, fecal incontinence in 39%, and sexual dysfunction in almost up to 80% of the cases. And you may be wondering, what are these surgeons doing to have these side effects so high? what these surgeons are doing is they're causing autonomic nerve damage, because the autonomic nerves are the tiny nerves around the mesorectum that are responsible for providing all of these functions. Why are they damaging them though? They're damaging them because they're invisible. So if you're looking at an MRI scan of the region, this is one of a patient also that is going in before the surgery. Here you can see the rectum, the bladder. Here you see the mesorectum, this fatty tissue around the rectum. But what you cannot see is the nerves. And the same is true for the surgery. During the surgery, there's no way to see where the nerves are. They're basically too small to detect by the human eye. So now here comes our goal in this project. And this goal was to model anatomical knowledge of where the nerves occur. And we wanted to use this knowledge that we then model to enrich the medical imaging data to do better surgical planning. So our goal was to end up with something like this. You still have this preoperative MRI scan of the patient, but now you have it enriched with the regions where the nerves are occurring so that the surgeon can be more careful in those areas. So you may be wondering, I said before that these nerves were invisible, but are there any people out there who know where the nerves are? The answer is yes, there are, and these are called anatomists. So anatomy is basically the study of the structure of the human body, and they have really a wealth of knowledge of exactly where these nerves are occurring. They're doing things like histological analysis, cryosectional analysis, and all of these things. Many papers exist on where these autonomic nerves are. The only problem is bringing this information into the patients. So to this end, we wanted to build a single atlas of human anatomy for the pelvic region, where all the surgically relevant information is available. So for this, we needed to build a basic virtual model, which we did based on the most high-resolution medical imaging data available at the time, which was cryosection. And cryosection is an imaging modality that you can unfortunately not do for live patients because it involves freezing the body and shaving off tiny slices of it and taking very high resolution images. So the resolution of this image is now 5616 by 2300 pixels, and we have 911 of such slices stacked on top of each other. This resolution is far higher than what you might find in any medical imaging data that can be done in live patients. So the in-between slice resolution is only 0.2 millimeter. What we did next is we had a medical expert manually annotate all of the important regions in this pelvis. So here's what that looks like. For each of these 911 slices, she manually outlined where the important surgical structures are. And this was a very important process because it allows us to reconstruct a 3D model from this. And this we call the 3D virtual atlas of the pelvis. So simply by stacking the segmentation slices on top of each other and doing a mesh reconstruction over it, we're ending up with this 3D model. Now, this 3D model is still not related to any patient, but it does show exactly the zones where these autonomic nerves are, here visualized in yellow. Already on its own, though, this is a valuable research for medical students because the anatomy of the pelvis is actually pretty complex. The pelvis is a funnel-shaped bony structure in which many organs are kind of squished together. So understanding the 3D relations is rather difficult. To share the knowledge that we already constructed here online, we built a browser-based tool for medical education where we, in the browser using WebGL, are visualizing our 3D model, and we're linking it to 2D imaging data. So here you see this cryosectional data once again. And we have a linked representation that also shows you the 2D slices within the 3D model. You have the option here to look at either the segmentation data, the original data, or both together. And this here really shows the relation between how anatomy may present itself in 2D slices, as you would see in medical imaging scans, with the relation to how it shows in 3D. Now you can easily turn off groups of structures or show them. And this was then meant as an educational tool for people to learn more about the anatomy of the pelvis. It's still online today, and better than that, it's also being used. So this model is used in the so-called Massive Open Online course, or a MOOC, on human anatomy on Coursera. And in the first run that we had with it, over 18,000 participants worldwide were using it, which was for me at the time also a great result because As we know, many of our research does not make it into practice, unfortunately. And this was one of the few examples for me that actually made it into something practical. I took another look at the course today, and it's still going strong. This year, already 40,000 students are enrolled, so it still has quite an active user base of people actually looking at this tool. Still, this doesn't bring us any closer to helping these patients with their rectal cancer treatment, right? Because that was the goal of our project. For this, we need to have a personalized patient-specific model. And one way of doing that is to register this atlas that we have built to the MRI scans of an individual patient to build this patient-specific model. So what we want to do is something like this. We have this 3D virtual atlas that is quite general. We have a medical imaging scan of a patient that is very specific, and we want to map the two together. For this, we need a process called registration. And registration is the process by which you have two volumes, one target volume and one source volume, and you want to deform the source volume to fit the target volume. Now, at the time, we were looking at what type of registration software is already available and if it could help us solve our task. And we found out that typically only 2D visualizations of the registration results are shown, or there's transformation tools missing to do this complex registration. Also, we found that these tools were not always suitable for non-image processing experts. So this registration process is rather complex. And if we wanted to have any shot of clinicians actually doing this registration of the atlas to the imaging data, we needed a more simple framework. So for this reason, we came up with this open source registration framework. And here you see two volumes. On the left, you see the target. And here you see the moving volume, the one we want to map. In this case, you're looking at a lung scan, by the way, it's not the pelvis. But what we did is we made many simple visualization options, like cropping, direct volume rendering. And what we wanted to do is to provide really simple tools to do this alignment, this registration. So here we're using some box to move one of the lungs around. And in the middle, you also see always the current outcome of the registration process. It's also possible to place pairs of corresponding landmarks in 3D if you can easily find them. Because if you place multiple of these pairs on the volumes, you can do a mapping based on that, right? So here in the center, you still see the result being updated the more landmark pairs we place. And this provides us with a pretty good initial registration, but to do a really accurate registration in our case, we need to do something deformable, right? To actually fit the organs of the patient. And for this reason, it's linked to a command line toolkit called Elastics, which has very advanced imaging registration techniques. We also provide a 2D visualization where you can see again the target volume and the moving volume, and here again the current registration results. So back to our goal, we didn't want to look at lung data registration, but rather at this atlas. Actually, it's not so straightforward to register an atlas to patient-specific MRI data. And the reason for this is that the modalities are very different. It's not like two CT scans that you can easily map together because the intensities are the same. So we needed to do a couple of tricks to make this work. And part of the tricks was that we needed to do some other representations in between to make the registration work. So starting out, we have our MRI data and we have our atlas data, which is basically a labeled volume. And what we can assume for the registration for our ends is that while the shape of organs vary, the distance to the zones in which the nerves reside is the same between patients. And we rely on this assumption to do the registration appropriately. So first, we segment the major organs in the MRI that we can see, and then we register the atlas organs to these. So we segment the organs and we end up with a label volume from the MRI as well. Remember, we cannot see the nerves, so we can only segment the organs and not the nerves themselves. We select these same organs from our atlas volume. Then to aid the registration process, it doesn't work so well with binary label volume data, so we calculated two distance fields. Because many of these registration methods rely on the gradient information, the normal label volume doesn't work so well. Then we register the two distance fields together. and we end up with a deformation field. And this deformation field is the one that would deform the distance field of the atlas to fit the distance field of the patient. Finally, from the atlas, we also select these risk zones, the zones in which the nerves are, and we apply the same deformation field to these atlas nerves. And in this way, we can really map these nerves from the atlas to the patient MRI. Now, this was rather dry explanation of it, but here you see it in action. So of course, these are 3D volumes, but I'm just showing you a 2D image to make it a bit simpler. But here we have the patient-specific image of an MRI. Here we have an image of the atlas at a similar height. And you can see they're not matched very well. But after we do the registration, we see that all the major organs are forming to fit the organs of the patient, especially the bladder here is quite an interesting shape. And you can see that the atlas bladder is also trying to fit this. Now, there are some errors in our registration, but what we care about mainly is that these outlines are correct, because that's where the nerves will be. We've done this now for 5 patients. So these are 5 real patients going in for this surgery. And doing that, we end up with custom label volumes for each of these patients. Now, this means that we can also make patient-specific 3D models rather than just a generic atlas model that we have. And here, it becomes a bit challenging to do a good visualization of these patient-specific models. Because what is a good visualization depends on what is surgically relevant and what is interesting for the surgeon to look at. So we discussed with the surgeons what they actually care about during the surgery. First of all, this is of course the tumor, because that is a structure they need to remove. Then there's the organs that they use as context, such as the mesorectum, the vagina and uterus, the bladder and the pelvic bones. And finally, of course, these autonomic nerves that they don't want to damage. Now you can categorize these structures as target structures. These are the structures that need to be removed during the surgery. Context structures, structures that are only serving as orientation, and risk structures that we don't want to damage during the surgery. Also, to the surgeon, the distances are quite important. So the distances of the nerves to the mesorectum, of the tumor to the mesorectum, and also of the tumor of the anus, because this actually determines the surgical approach that they're about to take. Something that the surgeons didn't mention they cared about, but that we said they should care about, is the confidence in the registration outcome. Because we're trying to do this registration as good as possible, but at the end of the day, there will be some inaccuracies where the registration didn't quite work so well. And we think it's important to show this also in the visualization result. So how do you visualize all of this together? The answer is not quite like this, right? Because here you don't have a good view on the structures that you care about. So here the mesorectum in green is not very well visible because other structures like the bladder here in orange and the vagina are blocking the view on it. So we needed to come up with some new surface visualization techniques to do this well. So for the context organs, if we visualize them as regular surfaces using Funk shading, you cannot perceive any structures that are hidden behind other structures. This is obvious, I guess. So here you have the bladder and the vagina behind it, and large parts are blocked. If you make them transparent, you can see more, but what you cannot see now is which one is in front of the other. So you lose your depth perception. By applying illustrative techniques like hatching, you can see a bit more of the shape. We also use the ghosted view to give a better impression of the shape, but still you cannot see which one is in front. Then if we add a silhouette shading, now you can see actually which one is in front while you can still perceive the shape. So here we have the best of both worlds. You can see the shape and you can still see which one is in front. Now for the distance visualization, we went with something rather simple that the surgeons are also comfortable with, which is mapping the distance to color. So in this case, we see the mesorectum here, and in red, the nerves are very close, and in white, they're further away. And we also added some ISO lines to make the shape more easily perceivable, as well as the distances. Visualizing them together with the risk zones will look like something like this. Again, we have added this ghosted view to make it transparent, so you can always look at the mesorectum underneath. We also added some lines here, which are helpful to indicate the height of the nerves in relation to the body. So here's what the combination of these visualizations look like. So here all the contact organs like the bladder and the vagina and the bones are visualized in the style that I mentioned before using this hatching and silhouettes. And the mesorectum is visualized with these color mapping on top. It's also important for the surgeons to relate the 2D imaging data that they see preoperatively to the 3D virtual model that we have. And for this reason, we again created a linked 3D and 2D representation. So what you have here is as soon as you click on any part in the 3D model, a 2D slice of the MRI scan will update to also show this part of the body. And this MRI slice is now also enriched with these outlines that were segmented, as well as the outlines of the nerves that were mapped from the atlas. So here we have enriched medical imaging data with the data from our model. Now back to this registration result confidence. For the registration, we wanted to do a sort of sanity check. So how well do the registered organ borders of the atlas now match the MRI organ borders? That was our main question. To compute this, we calculated the scaled distance for every MRI organ border point to the nearest atlas organ border point after registration. So here we end up with a 3D volume of distances. And here, if these are very, very unaligned, you end up with large values, right? That means we're not very confident in the registration outcome. If we sample this field now at the organ border, we end up with a sort of confidence value of how well the registration went in a certain area. But now we have a bit of a visualization challenge because we would like to show this confidence in the registration outcome and the distance to the nerve simultaneously on the same surface. So here we experimented with several visualization techniques to show both at the same time. The first one we came up with was a halftone representation, where in addition to the color mapping where we showed the distances, we also now show the confidence in the registration outcome. And here a bigger circle means we're less confident. So that means the values that are visible here are maybe not so accurate as the values visible here. We also tried similarly to show them a blended grid. And here, where the grid is blended in strongly, that means we're not so certain, and where it's not, we're very certain. In addition, we also tried to do a visualization based on the ISO lines that were already in the visualization. Here, if we're not confident, we make the ISO line very wide. And if we're very confident, we make it narrow. Similarly, we can do something with blurring. And we blur the ISO line where we're uncertain, and we make it narrow where we're certain. We showed these 4 visual encodings to the surgeons, and they didn't have very strong preferences, but they did prefer the halftone representation because they found it the most easy to interpret. The ISO lines one were a bit more difficult for them. Now we showed both distances and confidence in the registration outcome, but what we did not show was the distance to the tumor. Similarly, here we can also use this color encoding to show on the mesorectum surface directly how close the tumor is. Because between patients, the location of the tumor can vary quite widely. So here we have one that is quite in front and in the middle of the rectum. And here we have one that is quite low. But where is the tumor itself, you may ask, because it's of course inside of the mesorectum, it's not visible. So for this, we came up with an unfolding representation where you can pick a splitting plane and then kind of like the pages of a book or the layers of an onion, depending on how you want to see it, you can unfold the structures one by one. This works well because the pelvis is of course symmetrical. After we have unfolded, you can see the tumor in the middle, and you can also see the relation to the mesorectal wall. So in this patient, you can see that it's quite close to the frontal section. And you can also fold it back together once you're done. Here we see the same patient again on top, but in the bottom we see another patient. And here we see that the tumor is located very distally, so downwards in the patient, and towards the back wall also. And this type of information is very interesting for the surgeon to know about before going into the surgery. In the end, we linked all of these things together in a prototype surgical planning application. So first we have the MRI view here on the top right, and it's linked again to this personalized 3D model. try to skip ahead in a bit in the video, but it's not happening, I don't think. It's a rather slow video, but now it appears to be stuck, is it? Sorry, I'm having some technical difficulties with the slides here. Yes. Anyway, while the video is playing, I can also talk about it. So we evaluated this prototype with several surgeons. And what we found out was that experienced surgeons have a pretty good mental model of where the nerves are. And they built this mental model through years of experience doing these surgeries. And once they have such a mental model, such a tool is not so useful to them because they can already tell where the nerves will be. This is kind of obvious information to them. Where they saw the main value is in the tool is not to do it for every patient, but for patients where the situation is very complex and you want to do some extra preparations before you go into the surgery. And in addition, they thought it would be very valuable to have this type of tool when you're in training. So when you're studying to become a surgeon, because this can help you build your mental model. At the end of the day, I have to say that this was not really put into clinical practice, unfortunately, because this is a prototype a research prototype. It's not yet useful to go into clinical practice, unfortunately. But it did give a prototype of what is possible by mapping models to enrich medical imaging data. Yes. So, in general, in medical visualization, there are several trends and challenges that we can observe as things are happening. So there's advances in medical imaging that are also now challenging the things that we have developed in medical visualization research. First of all, there's a trend to go from static data to dynamic data. For instance, here we have 4D real-time stream data from an ultrasound machine. And if you want to do 3D visualization on such data, this is more challenging. Yes, there's also advances in data acquisition. So as we're going on in time, the machines are getting more and more powerful, and we're getting more high resolution imaging and also high throughput imaging, which makes it also more challenging to visualize. So for instance, the latest MRI scanners are having way stronger magnets, and this leads to increased spatial resolution as well. There's also a trend to go from visualizing anatomy to visualizing actual physiology. So how the human body is working rather than how it's just structured. And here, what they're doing is they're acquiring more and more multimodal data, as I was showing in the beginning of my talk, and also more multi-skill data. And it's a real challenge to integrate this multimodal data visually and to go across these different skills. Also, there's a trend to from going through looking at individual scans to looking at entire populations in cohort studies. And for instance, this could be based on electronic health records or on specific studies that also include medical imaging data. And here there's still many open challenges also for medical visualization. Finally, there's this research in computational medicine where they want to aim at building personalized simulation models to really cater the treatment to the individual patient. And in this talk here today, I've also shown you several examples of this. So the first example that I've shown, which was my brain, we're trying to go from static to dynamic, right? So rather than having a single 3D imaging volume, we're now having 100 of them. And it's not only showing anatomy as it was doing before, but it's also showing physiology. Also, the second example I showed with the pelvis is an example of a personalized simulation model where you're really trying to build a custom model to visualize the individual patients. So in conclusion, I've presented some examples of model-based visualization for anatomy, education, and surgical planning. Because if we want to aim at this P4 medicine, this predictive, preventative, personalized, and participatory medicine, we need to do more than just show a single imaging scan. So in this case, we wanted to predict where the nerves will be, prevent nerve damage, and also personalize pelvic models. And I believe that many challenges still await for us as medical visualization researchers. There's this increasing complexity and amount of medical imaging data, which places higher demands on analysis. And it's not just the medical imaging data that needs interactive visualization. There's also a lot of clinical data, a lot of gene expression data, and it's a real challenge to do any sort of integration on this. And I think for this, many more exciting topics in medical visualization can be explored. And I would also like to close with the fact that I think that medical visualization has the potential to kind of bridge these different subcategories we have as a field. So SciVis, visual analytics, InfoVis. I think medical visualization needs to go across all of this. And even if in the past I focused mainly on the SciVis aspects of medical visualization, I would like to integrate also more spatial and non-spatial data visualization because I think this will be very interesting. I'd also like to acknowledge the funding that I got to do this research and all of the awesome collaborators that have worked with me so far. And in closing, I'd also like to thank you for your attention. Thanks.
Thank you very much, Noeska. So we have time for a couple of questions. Who wants to go first? Klaus?
Hi again. You said your prototype of the pelvic atlas, where you can map the patient data with the atlas, is not in use yet.
No.
Do you think it is due to the constraints in the hospital, so you get the data from the radiographer and have to input it into the tool, and that this is too complicated?
Yeah, so there's a lot of processing involved indeed to get this personalized model, right? So you need to do the registration, and you need to do some mesh reconstruction. And I think another aspect of why it's not in use yet today is that you would need clinical validations before you're actually allowed to use any of this in clinical practice, right? So this is also a long and difficult process for which you need clinical trials, for instance, which is not something that we have the time and, let's say resources for, It would be perfect actually if a company would like to pick this up and continue with it. I think that's the only way to really get it into practice. But for this case, it didn't happen. Yeah.
Okay, further questions? Yeah, Patrick?
Nice talk, by the way.
Thanks.
And I wanted to ask for the nerves to visualize them. I think in the brain you can visualize the nerves with the diffusion tensor imaging or something like that. And
I'm sorry, could you repeat? I didn't quite catch that.
tensor imaging, diffusion tensor imaging, DFT, yeah, DTI, sorry, diffusion tensor imaging.
Yes.
I
Yes.
think I saw from the brain where you could visualize the nerves with this technique. Is this possible for the nerves around the rectum, or are they too small to really get the detection of the nerves?
Yeah, that's right. So DTI in the brain is very often done. And here you have these large nerve bundles that are kind of running together, which is why it's possible to do that. In the autonomic nerves, it's unfortunately not like this, and they're a lot smaller. So DTI so far has not been able to show the autonomic nerves either. Yeah. You would be able to see the bigger nerve bundles, but those are anyway visible during the surgery. So yeah.
Okay, then I would like to continue with the question. So, if I understood correctly, the atlas you created was then basically a surgeon labeled the data, and then you created one prototypic atlas.
Yes.
And then you have this uncertainty later on because patients are very different. If you have a tall guy and if you have a a small kid or so, have you thought about having an ensemble of labeled atlas, like a collection of atlas objects that you could use later on to reduce the uncertainty? And
yes. Yeah, that's a good and valid question. In the beginning of the project, it was also intended that we would keep enriching the atlas with more and more data, so more and more labeled volumes, so we would be able to do better registrations as well. In the end, we ended up with going by the assumption that we can use the registration to take care of most differences. So any differences in shapes of the bladder are taken care of by the registration, for instance. But what is still definitely valid is that there's huge differences in how people are shaped just based on also ethnicity, size, age, and all of these things. And especially gender is a big one also in the pelvis. So now we only have a female model. It would be fantastic to also build a male model to use on male patients. Yeah. So it would definitely be valuable to add more models to the atlas. But currently we're basing ourselves on one. Though the nerve zones themselves, they come from many histological studies. So these are based on also several people. Yeah.
Okay, thanks. And the second question was related to the application. Like the major application now is to use it for planning and also like educational purposes.
Yeah.
You also thought about like providing support during the surgery, or is that something surgeons don't want to have? Like using some kind of immersive technique, whatever.
This is something that the surgeons would love to have, because currently what they're doing often is they're taking either an anatomy book into the operating room, or they're bringing the static medical imaging scans that they have from before. And it would be absolutely perfect if they could really map this imaging data that they have from before onto the surgical side. What is the most challenging here is the registration. So for orthopedic procedures, this works very well because you can have like landmarks that don't change during the surgery, and you can map based on this. For these squishy kind of tissues in the pelvis, there's simply too much deformation to do a good job at the registration of the preoperative situation to the peroperative one. Yeah.
Okay, that makes sense.
Yeah.
We've learned from our collaborators at Imperial College, so we work together with Bernhard Kainz and the imaging group there. I think you know Bernhard.
Yes.
And I think they made some progress with registration using deep learning recently. Have you looked into that already, or?
I have not, but my collaborators in medical image processing from Leiden are also currently looking into this. So it seems that even transfer learning works very well for registration. So if you're learning from registrations on a certain type of data, let's say the lung, you can apparently transfer this to other types of data, which I find very interesting that this is even possible. But yeah.
Oh, I didn't know it. Okay.
Yeah.
Okay.
It's like transfer learning for medical image segmentation, but then applied to registration. Yeah. Yeah.
That's cool.
Yeah.
Any other questions?
Do you think that the surgeons would acknowledge additional visualizations like we have in other vision analytics systems? So, or do you rather stick to their 3D model?
So in my experience, I found that surgeons are rather open to new visualizations. So radiologists are perfectly adapt to looking at 2D slices and forming a perfect mental model based on this. So they're not the ones that would benefit so much from 3D, for instance. But the surgeons are really thinking more in 3D, let's say. So for them, these kind of more advanced 3D visualizations really help. And I think also the same is true for any sort of additional visual information that we can provide. I think that would be fantastic, actually, if we could do a combined visual analytics, sci-vis kind of approach and test that out with surgeons to see how that helps. Because in this way, you could also present more quantitative information that you found. Because right now, it's all kind of qualitative visualizations of the distances, right? Yeah.
All right, last chance to throw a question at Noeska. If not, then let's thank Noeska again. Great talk. And this brings us to the end of our lab talk series. We will have the summer break now, and we're going to restart then in October, but we don't know the speaker yet, but we will send it out via email. Thanks again. Have a nice day.
Thank you.
Enjoy working.
Thanks.
Bye.